DNA replication is the process by which a cell makes an exact copy of its DNA, ensuring that genetic information is passed on to the next generation of cells. It begins at specific locations on the DNA molecule called origins of replication, where the two strands of the double helix are separated by an enzyme called helicase, creating a replication fork. Single-strand binding proteins keep the strands apart, while another enzyme, topoisomerase, relieves the tension ahead of the fork. Primase then adds a short RNA primer to each strand to provide a starting point for DNA synthesis. DNA polymerase attaches to the primer and begins adding complementary nucleotides to the exposed DNA strands, continuously on the leading strand and in short segments called Okazaki fragments on the lagging strand. These fragments are later joined together by DNA ligase. The result is two identical DNA molecules, each consisting of one original strand and one newly synthesized strand, following the semi-conservative model of replication. This precise and regulated process ensures genetic stability across cell divisions.